Hi, everyone. I'm Bob Hastings, your host for the Society for Biomaterials Business Plan Competition, the 2021 edition. Thank you for joining us. Just some ground rules before we get started. The winner of the competition goes home with $1,000, and second place takes away $500. All the presentations have been pre-recorded, but there's a live Q&A session at the end where the judges can interact with the presenters, score each plan. Once I tally up the scores, I will announce the winners. We have 5 exciting ideas that will be presented today. We will start out with Trey Welch and Tremedics presenting a biodegradable stent for congenital heart disease, followed by Ram Saraswat and Cellfield Technologies LLC, a modular cell culture platform for preclinical testing. This will be followed by Chen Yang and Formocyte Therapeutics, an immunoprotection device for hypoxia reduction in cellular therapy, followed by Kelsey Collins and Metastatic Precision, a rapid diagnostic test for canine lymphoma, followed by Vinayak Mishra and Piezomembrane, a biodegradable piezoelectric surgical mask. We have 5 esteemed judges with us today: Stephanie Steichen from DuPont, Eric Sussman from FDA, Jim Curtis, formerly of Dow, Kip Thacker from Lifecore Biomedical, and Larry Thatcher from Tesco and Associates. So let's get started.
Good afternoon. I'm Jamie Wright, and I'm a postdoctoral researcher with Tremedics, where we make bioresorbable stents to treat congenital heart disease. So imagine that you've just had a brand new baby, and the doctors come to you and tell you that your baby has a congenital defect called coarctation of the aorta. And what this means is that within the first year of their life, your baby's going to need to undergo surgery. And in addition, after that, probably, as your child ages, they're going to require multiple procedures just to keep them healthy. Now, at Tremedics, we want to advance treatment for coarctation of the aorta. Our vision is to make it safer and easier to turn sick kids into healthy kids and to keep them out of the hospital. So coarctation of the aorta is just a narrowing or a pinching of the aorta. It restricts blood flow and causes the heart to have to work harder. And left untreated, there can be severe consequences like aortic rupture, aortic aneurysms, premature coronary artery disease. In addition to this, if these patients go untreated, they will experience heart failure or death by the time or before they reach their early 30s. So the current treatment strategy involves the patient first undergoing surgery within the first year of their life. Now, unfortunately, although the procedures are effective, the majority of the patients still experience a re-narrowing of the aorta within a few years. So within 4 years after the first surgery, typically these patients are requiring a metal stent to be implanted to hold the aorta open. Now, this metal stent is permanent, and it does not expand with the child as the child grows. So as the child ages and grows, the child will require multiple stent redilatation procedures to force the stent to expand and match the growth of the vessel. Now, each one of these secondary procedures, stent implantation and stent redilatation procedures, carry some risk. So stent implantation procedures carry the risk of possible incorrect positioning, possible migration of the stent. There could be tissue ingrowth, thrombosis is possible. In addition to this, stent redilatation procedures that force the stent further and further open can cause these stents to fracture. And if these stents fracture in the wall, it can lead to damage in the vessel wall. So imagine that we could eliminate all these secondary procedures and the risks associated with them, and instead implant a bioresorbable stent from the beginning. Now, this bioresorbable stent would hold the vessel open initially, allowing the vessel wall to remodel, and then eventually degrade, leaving the vessel free and unhindered to grow with the child. TradeMedics has designed a bioresorbable stent specifically to treat congenital heart disease. And in long-term animal studies spanning 5 years, what we've seen is that we have been able to correctly position the stent. And over the course of the study, the vessel has shown no restenosis, no thrombosis, no vessel occlusion, and only minimal scarring. So these stents do provide promise to reduce the current treatment for coarctation from multiple procedures to a single procedure. So again, our solution is Elusicor, which is a PLLA coiled stent with opposing coils to give it strength. It's covered by 4 patents. It is bioresorbable. It's balloon mountable and deliverable by standard transcatheter methods, meaning just like a metal stent. Now, we have filed for breakthrough device designation with the FDA, and in previous experiments have seen no late stent thrombosis or in-stent restenosis with the stent. It is MRI compatible and scalable to the diameter sizes needed for pediatric patients. Why Elusicor? So one of the biggest advantages of Elusicor is that with Elusicor, we've been able to achieve very flexible sizing. So we've been able to size this stent anywhere from 1.5 millimeters up to 20 millimeters and maintain radial strength while doing this. So the pediatric sizing requirements are usually range from about 6 millimeters to 14 millimeters on average, which is right in the middle of that range. In addition to this, we've been able to embed drugs in the stent, and the stent has not inhibited vessel growth. So looking at the competition, the CP stent and the Palmaz Genesis, the biggest competitive advantage that Elusicor will have is that it's made from PLLA and will resorb fully within the vessel, leaving the vessel free to match somatic growth to grow unhindered with the child. In addition to this, it is MRI compatible and can be implanted under MRI, which could reduce the radiation needed during the procedure and eliminate the need for a contrast agent. Now, short-term anticoagulation therapy would be needed when the stent is implanted. So globally, around 120,000 coarctation stent procedures are performed every year. This gives a total available market of about $810 million, which in this market is growing by 5% annually. In addition to this, once we penetrate the target market of aorta, looking over here at the stent markets, you can see that we can pretty easily expand our indication of use to include other areas like pulmonary artery, renal artery, peripheral arteries, and airways. The value proposition for physicians is that this is a minimally invasive surgery. And because the stent is implanted the same way a metal stent is implanted, no additional training is required. Now, for patients, there are fewer reinterventional procedures needed, and recovery is much faster when compared to surgery. So recovery will change from 10 days down to 1 day, and the cost reduces from $288,000 to $121,000. In addition, MRI would still be available to these patients even after stent implantation. Hospitals, this would lower the overall cost by 66%, and in addition, free up the catheter suite for other procedures. The regulatory pathway we would pursue is through a humanitarian device exemption pathway, which would involve first having the stent designated as a humanitarian use device, after which we could apply for marketing authorization through a humanitarian device exemption. Once we get the HDE, we could directly market to clinics where the device could be used under supervision or oversight of an IRB. In addition to this, we would be required to collect post-marketing surveillance data, and this data could eventually be used to convert to a PMA. So looking at the milestone map, you see that we've had a prototype. IP has been issued, and there's been a design freeze. In addition to this, we've shown preclinical feasibility with a long-term 5-year rabbit degradation study. We've shown preclinical validation in a pig model of coarctation. So right now we are seeking seed funding for contract manufacturing and the HDE submission. And for contract manufacturing, we're looking for biocompatibility, packaging, and sterilization testing. After all this, eventually we will move into post-marketing surveillance where we will require Series A funding. So for fundraising, the fundraising to this point has come from grants and from the founder in the amount of $570,000. We are currently seeking seed funding in the form of convertible notes in the amount of $1.4 million to cover contract manufacturing and HDE submission. And eventually, when we move into post-marketing surveillance, we'll need Series A funding. So what we're asking for right now is $1.4 million in convertible notes to cover contract manufacturing and the HDE submission. So looking at the financial projections, $1.4 million would get us through 2022, would get us through contract manufacturing and through an HDE submission before we would move into post-marketing surveillance requiring more money in 2023 and beyond. However, in 2023, if we were able to sell units, our break-even would be at 240 units. So the team, this is the team. I'm Jamie Wright. Trey Welsh is the owner. We've got Dr. Reddy, Dr. Forbes, and Dr. Gulasarian as advisors. We're a member of Tech Fort Worth. Collaborators include the AHA, the NIH, UT Southwestern, and Children's. Possible acquirers include Braun and Medtronic. And our mission is to turn sick babies into healthy babies. Thank you.
Hello, my name is Ram Saraswat, and I'm a fourth-year PhD student with the Nanoscience and Nanoengineering Department at the South Dakota Mines. I'm here to represent Cellfield Technologies. Cellfield Technologies research is focused on developing novel engineered biomaterial tools that could help scientists at pharmaceutical companies develop the first ever therapeutic drug to cure osteoarthritis, or OA for short. OA is a disease we get with age or injury. Currently, anybody suffering from OA is forced to undergo lifestyle changes or physical therapies. If not, they are forced to approach symptom-modifying approaches, and ultimately they're forced to undergo joint replacements. Unfortunately, this disease affects 33 million US adults and 500 million people globally. And there is no drug approved by FDA that could cure, heal, or prevent OA. And this is because there are insufficient mechanistic understanding of OA onset and development. There are no commercial lab tools that could model the multi-tissue nature of the joint in the early drug phase discovery. As a result, the pharmaceutical companies are predominantly dependent on small animal models for preclinical work. Unfortunately, the results from these animal models do not translate successfully in clinical trials. And finally, there is a high degree of clinical sample variability tied to insufficient mechanistic understanding of OA. To overcome this problem, we at Cellfield Technologies are developing a revolutionary product that gives us accurate information about OA using human cells. Our product, called Joint on Chip, will have different chips filled with different biomaterials for different cell types, and they will all be connected, as shown in the figure on the left, to study cross-cell talks or the effects of one cell type on another, either in hemostatic environment or under an OA-like situation, induced mechanically or chemically. In this figure, we are showing a joint-on-chip prototype that engineers the bone, cartilage, and the synovium. This will be our starting product, and as we grow, we can add more components of the joints to further advance our studies. Degradation of articular chondrocytes has long been considered as the primary cause of OA, and the Cellfield Technologies designed a product called CellWell, which is a novel engineered biomaterial articular cartilage model designed so that the chondrocytes do not lose their biological properties when studied in labs for a long time. The CellWell can be thought of a micro version of Ferrero Rocher chocolates, where the chocolate box represents the cartilage and the chocolate's chondrocytes. The cell wall will be used for culturing the chondrocytes on the joint-on-chip product. Both the cell wall and the joint-on-chip products have their patent applications filed and are now awaiting utility patent issue. We participated in NSF I-Corps in summer of 2020 and were able to identify our potential customers after speaking to over 100 potential representatives. And the ones in the slides are our representative customers we have identified so far. Based on what we learned during our I-Corps training session, we are continuing with our customer discovery process to identify more customers. During our I-Corps program, we realized that our customers would prefer us operating as a contract research organization, and our revenue model would be offering research services for a fee. And so we estimated our total available market as the global healthcare CRO market, estimated at $57.1 billion by 2025, with an expected growth of 6.8%. Our serviceable available market as the preclinical CRO market, which in US market alone was estimated at $4.1 billion in 2020, with an expected growth rate of 8.3%. And our serviceable obtainable market as the in vitro preclinical CRO market and arthropathy preclinical CRO market. Our joint-on-chip lab products arose from our customers' need for advancing the technology for getting a better understanding of OA using human cells at the laboratory. In our commercial journey, we identified our 3 partners: Innovate for ELISA systems, B2 Creations for additive manufacturing, and Biospherics for cytocentric physiological O2 systems. With the combination of these systems, nobody will be able to provide as accurate preclinical scientific data as a product can. We believe our cost structure to look like this, with high initial fixed operating costs, R&D and technological advancement costs, marketing and promotional costs, and working towards economies of scale and scope. We are currently working on validating these hypotheses. For our customer relationships, we believe to get our customers through Google AdWords, beta customers, conference and trade shows, scientific publications, and word of mouth. To keep our customers, we'll ensure to produce timely and reproducible results. And we will be customizing our product and adding more product lines to grow. This is one example of how we can grow our joint-on-chip product to further advance the technology. Finally, when cell-field technologies attains maturity, commercially, we believe we will have increased efficiency in providing reliable and timely results, which will lead to more successful OA clinical outcomes. This in turn will lead to improved population health and increased workforce resilience. In the future, we will be working on identifying more customers through conference and site visits. We have applied for NSF PFI grants, and in the future, we seek to apply for SBIR, STTR through NSF NIH. We will be working to ensure our filed patents get issued. If we win the SFB business plan competition, the award money will benefit the further development of our joint-on-chip prototype to help us bring this to reality. More importantly, though, the acclaim from winning such a prestigious award from such a respected society will greatly assist our efforts to attract venture capital funding and, most importantly, our customers. All this will help us in building a team of professionals that could drive the success of cell-field technologies. Thank you very much for listening to me today, and I'll be happy to answer any questions you may have.
Good afternoon, everyone. We are Team Pharmacyte Therapeutics from Nanyang Technological University in Singapore. I am Yang Chen, the team lead of Pharmacyte and a final year PhD student. Thank you for giving us the precious opportunity to share our technology titled Implantable Immunoprotection Device containing insulin-secreting cells with enhanced survival for diabetes therapy. Diabetes is a global epidemic with approximately 20 million type 1 diabetes patients requiring exogenous insulin injection. This leads to the current enormous global market of insulin pen, sizing of $22.15 billion USD in 2018 and estimated to reach $42.11 billion by 2026. However, with this standard of care for type 1 diabetes, 20 to 25% of patients still suffer from hypoglycemic attacks with associated fatality risk. To address this issue, transplantation of non-autologous islets into the portal vein of recipient to correct hyperglycemia has emerged as a potential strategy to treat type 1 diabetes. However, this method until now is still limited by lack of total availability and long-term use of immunosuppressants with adverse side effects. A cellular therapy that could prevent the risk of hypoglycemic attack and avoid using immunosuppressants will be able to address a large market worldwide, specifically targeting young adult type 1 diabetic patients experiencing hypoglycemia. The total addressable market for such technology will have an estimated annual revenue of $33 billion USD. Considering only the target markets like the US, where such treatment is affordable, the serviceable available market and service obtainable market will reach 82 million USD and 8.2 million USD respectively. The beachhead comprises of patients that normally require islet transplantation in the US, now adopting such treatment will yield an annual revenue of 1 million USD. Islet encapsulation has emerged to achieve these goals. Typically, islets are encapsulated in semi-permeable hydrogel microspheres to be immunoisolated before transplantation into the abdominal cavity, bypassing the need for immunosuppressants. However, so far there is no clinical success of encapsulated islets due to hypoxia-induced cell death. Several factors contributed to the hypoxia in the encapsulated islets. Firstly, the natural spheroidal geometry of islets is unfavorable for mass transfer, leading to poor cellular viability of islets. For example, this image captures a representative human isolated islet. Many red cells were detected in the interior of islets, demonstrating poor viability of islets. The second is the clumping of microspheres implanted in the abdominal space, hindering mass transfer. Thirdly, the conventional transplantation site of abdominal space has relatively low oxygen partial pressure. To address these challenges, Formocyte has developed a platform technology, which is an immune protection device for therapeutic microtissues. The device enables fabrication of toroidal microtissues like a donut to improve cellular survival by minimizing hypoxia. These toroid microtissues can be encapsulated in a homogeneous distribution in the device to prevent microtissue aggregation. Furthermore, this device allows the transplantation to subcutaneous site, which has higher oxygen partial pressure compared to abdominal space. A PCT patent has been filed to protect this technology. The proof of concept study showed that toroid microtissues exhibited significantly less dead cells than spheroid microtissues. Moreover, higher hypoxia signal was detected in uncontrollably distributed toroids than in homogeneously distributed ones, demonstrating that preventing microtissue aggregation will contribute to hypoxia reduction. We also confirmed that the encapsulated toroid microtissues can secrete insulin responsively to the glucose stimulation. In a preliminary in vivo test, we confirmed that the micro device can be transplanted into mice model and retrieved with mechanical integrity, demonstrating that our device can protect the encapsulated cells in mice model. Here, we presented the competitive analysis of our potential technology with existing devices. Our technology, along with Semma Therapeutics and Beta-O2, each possesses a key feature to reduce hypoxia, which Signal Therapeutics and Synover technology don't have. Furthermore, compared with Semma Therapeutics and Beta-O2, our technology has simpler device design without the need of any accessories. rendering it easier to be translated into clinical application. In addition to diabetes indications, our platform technology has the potential for treatment of other hormone deficiency diseases by encapsulating different types of cells to secrete missing hormones of interest accordingly for the restoration of physical balance. For example, hemophilia A, characterized by failure to produce blood clotting factor A, has a prevalence of 1 out of 5,000, affecting mostly males. The patient population is 20,000 in North America and Europe, with a market potential of 10 billion. In a preliminary in vitro result with our collaborator, Prof. Peter George, we confirmed the feasibility to fabricate toroid mark tissues in the Formosite device using HEK cells engineered to secrete blood clotting factor A. We expect Formosite device also has the potential to treat thyroid disorders, neurodegenerative diseases, and lysosomal disorders. Here is the 5-year milestones that we expect for the development of our technology and startup. In 2021, we plan to test Formosite macro device housing primary islet cells in mouse model. From 2022 to 2023, we expect to utilize human-induced pluripotent stem cells by collaborating with a stem cell expert. From 2024 to 2025, we expect to establish our startup and raise funds from VC to GMP manufacture macro device housing human-induced pluripotent stem cells and evaluate device efficacy in advanced animal model. After that, we will proceed to apply for approval for clinical trials. Our macro device can be classified as cell device combination product regulated by FDA's Office of Cellular, Tissue and Gene Therapies and Center for Biologics Evaluation and Research. For our long-term strategy, we envision the complete development and commercialization of our device housing insulin-secreting cells with type 1 diabetic patients as the main customers. In short term, we anticipate to license our device to biopharmas with interest in cellular therapies requiring such complementary cell housing technology. Alternatively, acquisition from such biopharma company will be our exit strategy. For type 1 diabetes indications, we expect to leverage the distribution network of biopharma companies as the channels to deliver our products to customers. To maintain the operation of the startup, we need to finance fixed costs including rental fee, IP maintenance, and manpower costs, and the variable cost of R&D activities and regulatory processes. We expect a revenue stream including product sale for type 1 diabetic treatments and royalty or licensing fee for platform technology from non-type 1 diabetic applications. Our team, Formosite Therapeutics, including an assistant professor, 3 PhD students, and an MBA student, was awarded with NTU's Multidisciplinary Team Project Fund for technology commercialization. Assistant Professor Dr. Chen is a biomedical engineer with expertise in cell-based therapeutics and biomaterials. She is an inventor of a US patent of another technology platform. I, Yang Chen, is the team's technical lead with expertise in designing microfabricated tissues and hydrogel-based immunoprotection macro devices. I was the winner of Nanyang Technological University's 2020 Entrepreneurship Award. Nam Chen is a biomedical engineer working on device prototyping and scale-up manufacturing. In addition, she is a biologist focusing on evaluating clinically relevant source of pancreatic beta cells. Arnold Giovanni is an MBA student in Nanyang Business School of NTU. Our industry mentors are Dr. Po, Head of Global Operations in Hedent, and Dr. Song, founder of Ternium Biosciences. We collaborated with Dr. Thiel, who provided us with human-induced pluripotent stem cells. Dr. Ali specialized in islet biology, and Dr. Liu as a senior consultant of metabolic diseases. Thank you for your attention. That's all.
Hi everyone, my name is Kelsey Collins and I'm the entrepreneur with Metastatic Precision. We are developing a new point-of-care diagnostic test for cancer that will provide veterinarians with results in minutes rather than days, while also reducing the costs and stress to the pet owners. And these are my team members. Dr. Berg is an endowed chair and professor in the Department of Small Animal Medicine and Surgery at the University of Georgia. And Dr. Taylor is a Chief Technology Officer at Polymed Incorporated and has 19 years of small business experience. And that's me on the right. I just graduated from the University of Georgia last spring with a bachelor's in animal science, and I have 7 years of experience as a veterinary assistant. And this was my dog, Daisy May. So here's a healthy x-ray that was taken a couple of months before her illness as a way of comparison. So in 2018, she wasn't quite acting right and appeared to have gained a little bit of weight in her belly area. So I brought her into the clinic to have some blood work done, and everything came back perfectly normal. The vet who did her exam told me that it might just be her age setting in and not to worry. But we did end up retaking an x-ray just to be safe, and sure enough, we saw that her spleen was enlarged. I spoke with 2 different vets who told me that there was no way to be certain if this enlargement was cancerous or not without doing surgery. Both of these vets actually suggested euthanasia at this point, just because the chances of it to be cancer were so high. Unfortunately, there are so many other owners who find themselves in this exact situation that I was in, and they decide to euthanize without even knowing if their dog has cancer or not. As you can see, having a splenectomy done is a very invasive and risky surgery. I decided to go forward with a splenectomy on Daisy, and I was really glad that I did, because to all of our surprise, her mass came back benign, and soon enough, she returned to her happy, healthy self. Until last July, I noticed that she had a firm lump on her mammary gland, and right away, I knew it was bad. We ran blood work just like before, and again, it came back perfect. I decided to put her through surgery, not knowing if it was cancerous or not, but this time it was confirmed to be a carcinoma. The vet who did her surgery explained to me that she got good margins and didn't suspect that the cancer had spread. We thought the surgery was curative, and she didn't recommend doing any chemo treatments. I just wish that there could have been some way to test her to know if her cancer really was gone, because 5 months later, her lymph nodes became swollen, and before I was able to get her into the vet, she passed away. I believe that she had lymphoma, but because I wasn't able to act fast enough, I won't ever have that closure. So my experience has motivated me to find a solution to this common issue. The medical problem that we are addressing is the lack of cancer screening and the time that it takes to receive a confirmed diagnosis. We interviewed over 80 pet owners last fall, and almost 30% of these owners never received a confirmed diagnosis. Current methods used to diagnose cancer in dogs oftentimes require an appointment with an oncologist, and clients are usually referred to more than one specialist for further testing. Delayed treatment time can be detrimental when dealing with an aggressive cancer case. And in many cases, by the time that the client receives a confirmed diagnosis, the cancer has already progressed and their treatment options then become limited. So our solution is a rapid in-clinic diagnostic test using only a fine needle aspirate sample, which are already done routinely in animal clinics. But instead of sending the sample off to a reference lab and waiting days to hear back, the vets will be able to use this test and have results in minutes. Our unique patented fiber design separates cancer cells from benign cells based on their physical differences. And the vet staff will be able to know that cancer is present just based on the distance that the cells travel up the fiber. So we are currently in phase 1 of our technology development. With Precision Vet DX, a veterinarian would take a needle biopsy of a lymph node or a tumor and then aspirate those cells into a mixture of saline. The cancerous cells would travel up the fiber bundle at a faster rate than the benign cells and then be isolated into a cap at the top. And after about 20 minutes, the veterinarian or a skilled technician could then take that cap and look at it under a microscope to interpret the results. In the next phase of development, we want to explore the possibility of creating a test that provides a clear, positive or negative result, so that any staff member will be able to interpret the results without the need for a microscope or any training. And once this becomes possible, this test may be more applicable to human medicine. There is currently a patent for this device, which was issued in 2017, and all rights are owned by the University of Georgia. Last fall, we conducted customer discovery interviews where we had the chance to speak with veterinarians, vet techs, industry experts, and pet owners, but we focused primarily on pet owners. And even though they came from all different walks of life, they all had one common belief, and that is that cancer diagnostics and screening need to be improved. So the time that it takes to be seen by a specialist and then having to wait for a biopsy result is extremely stressful for the owner, but it's also really stressful for that sick pet to be put through so many invasive diagnostic procedures because many of them require anesthesia. With current technology, the primary vet is limited to basic diagnostics like an X-ray machine or blood machine, and they oftentimes don't provide a clear answer. So that vet will usually refer to a specialist who is better equipped, and this is common especially in remote areas. But with our technology, the primary vet is going to be able to diagnose cancer in-house, which is going to increase profits and client satisfaction. The vet will be able to quickly know whether cancer is present in the animal during the same appointment time. And that's going to allow the pet owner more time to process the different treatment options. When a client gets referred to a specialist, it can take anywhere from 1 week to 1 month to be seen. And all this time, the cancer is still spreading. But with our product, the owner will be able to act quicker and get their pet into an oncologist to start treatments right away. Since our product will be the first of its kind, there really aren't any competitors yet. And we had to make the assumption that our products would fall into the same market as current cancer diagnostics, even though our product would be very different from current diagnostics, like an MRI machine or a flow cytometer. The target market for cancer diagnostics in companion animals is around $875 million, and around 21% of diagnostics are specifically for canine, which is around $184 million. Based on other products that were new to the veterinary world, we predict that it may take between 2 to 3 years for the majority of vets to adopt our product into their diagnostic protocols. The adoption approach in veterinary medicine is very different from human medicine. Many veterinarians will wait for a new product to be on the market for at least a year before they decide to try it. However, veterinarians will commonly try a new product if they learn about it in a medical magazine or at a conference, or read about it in published literature. With our understanding of how veterinarians make decisions, we will be working with key opinion leaders in the field to get pets and their owners the cancer diagnostic that they need. During our customer discovery interviews with veterinarians, we asked them, how often do you take a fine needle aspirate sample? And the average answer was between 3 to 5 times per week. So we used that number to apply toward our product. Our product will be sold in a quantity of 10 for $250. If we can achieve an adoption rate of 15%, our gross revenue will be around $35.1 million. After meeting with all these veterinarians, we know that proof of accuracy is the key aspect in getting a vet to adopt a new product. Right now, we're working on developing our minimum viable product and hope to have that finished by 2022 so we can start our soft launch. During our soft launch, local veterinarians will be able to try out our product and provide us with feedback while also still using current methods. This way, we can compare the results of our product to the results of current methods being used so that we can develop an accuracy percentage. The data we gather from our soft launch will be used as proof of accuracy for our product when we get ready to publish literature. We are asking for $200,000 to help with further development. Further funding will allow us to explore phase 2 of our development and help produce a successful minimum viable product. We are also asking for connections with small-scale or private veterinary manufacturing and distribution companies so that we can explore potential partnerships in the future. Unfortunately, Daisy's story is not unique. There are so many other dogs out there going through a similar situation right now, and some of them are as young as 2 years old. But with your help, we can make a difference to future pets by providing them with an earlier diagnosis and a better chance for survival.
Hello, everyone. My name is Vinayak Mishra, and I'm currently a senior in the honors program at the University of Connecticut studying molecular and cell biology. I've been working with Dr. Tan Nguyen, Dr. Yang Lu, and Tin Lee on our new product, a biodegradable and reusable mask. At the beginning of the COVID-19 pandemic, there were shortages of almost everything, but most importantly, masks. Since then, some of the largest manufacturers have stepped in and started producing massive amounts of masks to offset this deficit. 3M even started producing upwards of 90 million units a month. All of these masks have been disposable, one-time use masks, which have caused pollution to our land and most importantly to our seas, which are near impossible to clean. According to the United Nations Conference on Trade and Development, about 75% of these masks end up in the sea, polluting our ocean life. As the pandemic continues and we return to a new normal, new masks are needed, and they need to be environmentally friendly. Currently, we see 3 different types of masks: N95, surgical, and cloth masks. N95 masks are very efficient and can filter 95% of particles, yet are not reusable and definitely not degradable. Surgical masks are a little less efficient, but again, cannot be reused and are not degradable. We can see that the problem lies here. Cloth masks are reusable after being washed, but aren't very good at being masks as they can't filter much. Since the pandemic, many companies, new and old, have joined the market in trying to make reusable masks, but none are doing it like us. Our masks are not only provide higher filtration efficiency than surgical grade masks, but are also reusable and biodegradable. Our masks, as shown here, is constructed of a PLLA sandwich, which houses our PLLA piezoelectric filter. It is made of an FDA-approved poly-L-lactic acid biodegradable polymer, and its properties allow it to be sterilized and reused. Not only that, but our mask is very tunable. Its parameters can be changed in order to increase its lifetime, filtering efficiency, and much, much more. This new trapping technology allows our masks to have higher efficiency than surgical masks while also keeping it environmentally friendly. Our mask also employs a novel active trapping technology. When someone breathes with our mask on, our filter actively creates an electrical charge which traps viruses, bacteria, and other small particles. This is all done by our piezoelectric membrane filter, as can be seen here. After the mask is used for up to 5 days, it can be simply recycled, where it will never pollute our land or oceans. Currently, the industry for the mask in the United States is at a high of $1.4 billion due to the pandemic. As the pandemic fades away, this mask has a potential $8.1 billion market worldwide and a $900 million market in the United States alone by 2025. A study by Forbes done in the United States and Australia suggests that 77% of people want to live more sustainably. And my recent study here at UConn has showed me that 76% of UConn students would love to buy environmentally friendly masks over others. When we hit the market, we want to target these universities and young adults who are environmentally conscious. Our commercialization strategy entails launching our first product as soon as this year because it requires next to no regulations. After a successful launch, our team will look into FDA and NIOSH approvals so that they can be N95 rated and approved to be used in hospitals. This will happen once we improve the parameters for our masks and get it to the required filtration efficiency of an N95 mask. These will, of course, not be necessary at the beginning, as we will focus our sales to the general public only. We hope to optimize our in-house electrospinning machine as shown here, which will be able to produce multiple masks a day, and we hope to purchase more of these machines to expedite mask production. Our exit strategy as of right now is to IPO and expand our line of products to our other patented technologies, such as a biodegradable bioactive periodontitis membrane. Another option we are considering is licensing and selling our mask technology to a larger company in the industry, such as 3M. In terms of generating revenue, we will follow a mix of transactional and subscription business models. Our sales will be made directly to the public via our website. We will also have a dedicated marketing team to expand our customer reach through online and social media advertisements. Other than directly buying our masks, we will offer subscription plans to the customers in order to drive in recurring revenue while offering our customers guaranteed mask replacements on a monthly basis. Once we generate stable revenue, we will expand to getting a sales team to start working with hospitals, universities, and other large institutions and businesses on wholesaling our product to them. This will ensure that we are profitable with the general public and can ramp up production in the meantime to accommodate orders from larger businesses. So far, we have tested our filters and created a prototype of our mask. We are hoping to launch our company and the first product by the end of this year. As we stated earlier, we hope to start off with generation 1 of our product, which will be comprised of a cloth mask with our novel degradable filter inserts, and then move on to the fully biodegradable mask later. The funding we receive from this competition will help us secure a lab and manufacturing space where we will be able to de-risk the mask as well as start productions. These will ultimately help launch our first product design and generate revenue, which will be used to fund generation 2 of the masks. We want to provide mask protection in a greener way. Thank you.
Thank you, everybody. Those were some fantastic descriptions of ideas. And I'd like to open the floor to questions from the judges. So it looks like we have all of the presenters online, and we have the judges online. So I open the floor for questions. See how it goes.
I've got a question for the resorbable stent company, Trey.
Yes.
Could you give us some insight as to how this resorbable stent technology may be similar or different from that Abbott used and had to go back and do a redesign? And I'm just vaguely familiar with it, so yeah. I'm curious as to why you think this resorbable technology is going to be successful out of the gate.
So yeah, the stent is made of the same material, but the design is a little bit different. So the Abbott design and a lot of the other bioresorbable stents use a tube that's laser cut or cut in some manner, whereas ours, we have a coil design. So we extrude fiber, the fiber is drawn and annealed and processed, and then it's wound and we have an opposing coil design, and the opposing coils help to give the stent more strength. So it can maintain its, so it can keep the vessel open. So the big difference is for us, what we're seeing is that on all of the studies we've done multiple studies with rabbits, multiple studies with pigs. In all the studies, we're seeing endothelial coverage within 1 month. So that's the big difference.
Might add one point. A lot of these other companies that put these stents in, they have issues with malapposition of the stent being secured against the vessel wall. So what ends up happening is the stent's not getting covered by tissue for degradation and leads to thrombosis issues.
I question because you mentioned that it's different because the others are laser cut tubes, but there also is the Igaki Tamai stent, which is also a filament-based stent that had significant clinical issues, particularly for deployment. So there are predicates. You were speaking to specifically the question about the Abbott stent, and to that end, I look at histology. And what have you found in your histology regarding identification of the tissue that replaces the strut? That certainly there has been some issue with proteoglycan replacing that stent strut, and the ultimate endothelium and interelastic lamina being somewhat dysfunctional. So how does your stent, being made out of the same materials, elicit a different type of cellular response?
Yeah, so so we did full resorption within, I don't know, 2.5 years. It depends on what vessel we're in here. But in the aorta, 2.5 years, we're showing resorption. Again, one of the things we've tried to do is slow down the degradation rate. So by increasing the crystallinity in the stent, so post-processing the fibers allows us to increase crystallinity. It slows down the degradation, the hydrolytic degradation, and it slows down the adverse effects or how those byproducts affect the cells. So that's really the difference that we're looking at.
To more point out to your question, Larry, the resorption of the stent did show over a 5-year term some scar tissue formation, but we did not notice any dysfunctionalization of the artery in response to putting catheter wires in. We did see a normal type response, but we did not inject acetylcholine to verify that in our study.
So to the point of the Abbott stent again, that your product is proposed to be delivered to a vulnerable population, pediatrics, and what are you able to speak directly to the mechanism by which the Abbott stent did fail and why your stent overcomes that potential failure?
Yeah, Trey, if you want to.
Yeah, I can answer that. So the issues with the Abbott stent is they can't make a stent for pediatrics to go in the aorta. Their limitation size is to 3 to 5 millimeter. So we were able to overcome these size limitations. We've tested up to 10 millimeter in a pig coarctation model. But what we did find is we do get good apposition to arterial wall, leading to tissue coverage of the stent, which once we have tissue coverage, we reduce the risk of late stent thrombosis or any other thrombogenic events that can occur with leaving a PLLA-based stent in the blood vessel and blood contact for long term.
The late stage in-stent thrombosis 2 to 3 years post-implant of the Abbott was not in the presence of an unendothelialized strut, but of dysfunctional tissue, underlying tissue. When you did your porcine model, did you use a Yucatan mini swine? And for how long was your implantation?
It was a Yucatan swine. The implantation was up to 1 year in a diseased model, meaning they created a coarctation, then went in and implanted the stent over the coarctation, and a year later examined them.
Did you follow these with OCT?
We followed with IVUS.
With IVUS, okay.
We hope to have the data published this summer.
With the high expansion ratio there, was that as interesting as far as the significant radial strength that you might have to have in your specific indication? And I'm curious about the immediate recoil that you experienced.
Yeah, so the recoil is dependent somewhat on the stent size, but it's smaller stent sizes, 3, 4, up to 6 millimeters. It's less than metal, so percentages approaching 1%. After that, the recoil goes up a little bit, goes up to in larger stent sizes. Let's see, we got recoil of a little over 1%, 2% in that neighborhood.
I'm curious what your estimated cost of manufacturing would be for one of those stents.
Yeah,
the cost of manufacturing for one? Oh, sorry.
Yeah, so we've been negotiating with Medical Murray for this cost for manufacturing through a contract manufacturer. They're quoting us about $500,000.
As far as regulatory, I saw that you applied for breakthrough status, but I read through the guidance on breakthrough and I didn't see mention of humanitarian devices there. What's the crossover?
Good question. So you can apply for an HDE application. You can apply for the breakthrough device as well. The crossover is once you apply for the if you're awarded breakthrough device designation, and you run through a HDE, you lose you lose part of the program that you can take advantage of on a PMA application with the BDD. So through correspondence to the FDA, they told me I could do an HDE and I could still get a BDD designation. I just couldn't take advantage of getting putting in the queue quicker for discussions with the FDA for going through clinical studies.
Okay, that's interesting. As far as clinical studies, could you clarify when those are planned? From your submission, it looked like that you were going straight to marketing. I didn't see a plan for an IDE.
Yeah, so based off the HDE application, you can get direct marketing of the device. And then we start the IRB process with going through a clinic, and then we can begin our studies. So it'd probably look after HDE is approved. We're probably looking at 3 to 6 months before we could kick off a human trial.
So do you think, so that's interesting because the HDE itself requires some evidence that there's going to be a benefit to the patient. And so you think you'll be able to satisfy that evidence prior to conducting a study?
I think we can, but we're working with Proxima CRO, so they're guiding us through this process right now.
I wanted to ask about the financials that you mentioned. It looks as if you've assumed that you're going to get the same value outside the United States that you would get per stent in the United States. What's the basis for that?
So let me try and understand your question. You're saying that the same sell for the cost of the device outside the U.S. versus inside the U.S.?
Yep.
Is that what you're asking? Yeah, well, our cost estimates are based inside the U.S. So selling outside and expanding further into the EU or others, we'd have to negotiate with them. But we would like to achieve that same price point if possible. But we don't have those numbers yet.
I see. And do you have any examples currently where they've been able to get the same price point or potentially more? Because, United States prices are often as much as 6 times higher than European prices.
Correct. Yeah. So I don't know what the European pricing is. I just know what the pricing we pay here in the US.
And I suggest we move on to the second cell field with Ram, and any questions on that technology, please.
I was wondering if they could comment on the validation plan, as particularly as you're looking at this as a service and ultimately a fee for service reimbursement, and what that would, the validation plan would look like, and what regulatory requirements and hurdle settings.
So as of now, we will be more in the preclinical, and we'll be doing services, research services to the companies that themselves will be doing the clinical. So we do not see ourselves ever being doing any regulatory work on our end, because what we are doing is we are doing laboratory studies, and whatever research studies we get, we just sell them to these companies, and then they take care of the clinical trials or whatever happens with the regulatory process.
What evidence will you be able to provide of the predictiveness of your technology to the potential customers?
Sorry, could you repeat your question?
I'm sorry, I think I interrupted Stephanie there, but I was going to say, what evidence do you expect to need to provide to your potential customers that this is that this works?
So yeah, so this will be a lot starting from the scientific results. So in the joint, for every joint and for every cell type that's involved in the joint, there is a specific biomarker that these cells express. So there's a history of literature which explains that these are the good markers and these are the bad markers. So we will be starting from there, where for each cell type, we'll be showing that conventionally, if you put the cells on conventional platform, you get these bad markers. Or say for osteoarthritis, these are the bad markers which it's published in the literature, that they show these are the bad markers, but now you're putting in our platform and see these are the good biomarkers. So that will be the starting point. And then moving from what the customer says that, yeah, we want you to do these particular testings or do these analysis for us, so we can do those for them.
And just a quick follow-up, are there any examples of organ-on-a-chip systems like this on the market now that you're using?
So there is, specifically with respect to joint on chip, there is not. There is the closest is a professor called Rocky Chuan's group in Pennsylvania, and then they are currently doing some kind of joint on chip concept. But what their concept is entirely different from what ours is, and they are also not. As far as what I have read, they are not in the commercial field yet. They are still in the development phase. But specifically with respect to joint on chip, there is nothing out to our knowledge.
So what's the just one last one on this line of questioning, what's the best example of currently marketed organ on a chip product in the world, I guess? I would say the concept of organ on chip will be the one that I think it came from Boston University, where they were doing lung on a chip and heart on a chip. There is some new work coming for intestine on a chip. So those are in development. But commercially, I think it started with PDMS microfluidics, where they were doing lung on a chip. Thank you.
I have a question that is, Eric did steal my first question, so I'm going to sort of ask a follow-up to that. So he was asking about the predictability of this system that you could provide to potential customers. Do you have any plans as part of your business, your startup, to compare the results that you would get from this joint on a chip with something that you might get from whatever would be the gold standard animal model, let's say, or taking that one step further and what you would see putting that into humans. So I guess any thought of how you're going to validate that what you're seeing on your device is similar to what you would actually see in patients?
As of yet, I think our gold standard will be. And sorry, let me give you a little bit background. So the gold standard is animal models, but animal models do not translate well into the clinical trials. That was the whole reason of coming into this. Now, in the lab, we will have the traditional cell culture plastics, which is widely used in the lab, and we can use that as a control. Down the road, definitely at some point, we would include animal models, but at this point, we do not believe we have enough funds and scientific knowledge to do ourselves. Maybe we might do with like a CRO, for example, Boulder Biopath in Colorado. Boulder, they do a lot of animal studies, so we might do something collaboration with them. But as of now, our main controls will be the traditional lab culture plastics.
So as moderator, I have a question. I apologize. I'm trying to let the judges ask all the questions, but I can't help myself. So what is it? What is the thing that you plan to sell? Is it just the chip, or is it going to be harvesting technologies for the cells, or explain exactly the kit, I guess. Please.
So we are, so this is so we are not selling any of those. What we will be selling is, we will have all these products in home, but we will be selling the research data that comes out of conducting these experiments. So we will be doing the experiments, and whatever data we get, we will be selling that. And this was, again, so last year when we went into NSF course, we actually thought that we will be selling the chips, but we realized that these pharma companies don't want the chips. They want more of the research data, and they want us to do all those experiments ourselves in the lab.
Thank you.
How much work was done with the material of construction of the chip, the substrates on which the cells are growing?
So these, as of yet, the current model is actually worth 2 PhD theses at our school. So one is a person who is graduating this year, and the second work is what my PhD thesis is all about. So yes, we all did all this work ourselves in our lab, and now we are trying to commercialize this. We are still exploring, I would say, in terms of biomaterials, we are still exploring other possibilities because we believe we can always improve upon our material work.
So I guess what I'm curious about is, how do you make a decision as to what is a good material and what's a bad material for the substrate that you're using to create these chips?
Sure. So at this point, in terms of materials, so each tissue of the joint has a specific histology. And we actually did those studies to actually see, first of all, how they look in the body. And then inspired from those, we used the biomaterials. Now, when it came to the biomaterial selections, we were checking for substrate stiffness. That was one of the big parameters. Another thing was we were checking for opaqueness, because when we are, for us, because we are very big on imaging, so we want to make sure that these biomaterials are as translucent as possible. The thickness of the sample, because all these objectives through which we will be using, they have a limit working distance limitations. And then sometimes, like say, if we are, for example, there's a product called CellWell, where we actually are modeling the collagen 2 fibers. So now we went and when we were modeling the fibers, so we use polyvinyl alcohol fibers. And now we measure the diameter of our electrospun fibers, and then we compare that with the same fibers which are actually present in the body. And we found that those diameters were matching. So these were the parameters we were looking.
Thank you, Ram. I think we should move on and have any questions for Yang Chen and Pharmasite. Please, judges.
Is this device going to be marketed to all type 1 diabetes patients or a subset of type 1 diabetes patients?
It's designed for all type 1 diabetes patients, but it's majorly for those who fail to correct the blood glucose using the traditional insulin injections.
And so how will a physician determine whether that the specific patient is a good candidate for this technology?
Yeah, because some patients are very sensitive to the insulin, and the dose the physician prescribed to the patients may not be so applicable for each individual patient. In that case, the patients may suffer from severe hypoglycemia with the traditional insulin injections. So we believe that our cellular therapy products can the cells of can automatically sense the blood glucose level of the diabetic patients and then release the insulin in amounts that is just enough to fix the blood glucose, restore the euglycemia level without subjecting them to any to severe hypoglycemia.
Your reimbursement target is based on the current islet transplantation, IV transplantation costs. Yet, as a new device, you'll be confronted with some type of a cost-benefit rationale and ultimately some reimbursement obstacles. How would you propose to overcome those?
Sorry, could you repeat the question again?
Yes, you stated that your current pricing and reimbursement is based on islet transplantation, which is of an IV transplant, and you mentioned something like $145,000. So that is obviously procedure based, but as you're dealing with a new device, new treatment plan, how are you going to address reimbursement obstacles and cost benefit analysis?
Yeah, thanks for the question. Right now, the price, we estimate the price of our product based on the islet transplantation cost. This price will be a little high, but we are thinking the alternative strategies to maybe launch it in the countries with the higher ratio of the insurance scheme, such as the UK. Their insurance coverage ratio is higher, and then maybe it's more applicable. The medical scheme can be more applicable for this treatment, and then we will kind of expand this in other countries, such as and then we may once we open the market and then they see the potential of our products for the correction of blood glucose, maybe we can bring it, make it inclusive into the medical scheme of other countries as well.
You mentioned the UK with their Institute of Comparative Effectiveness. Really not sure they're going to be applicable to paying quite a lot more unless you demonstrate a huge advantage. And if you don't get it in the treatment plan there with their Department of Health, it's not going to fly. So I wouldn't cite the UK among the list of countries that you should be pursuing first.
Question a little bit more on the use side of your technology. So you were talking about implanting this subcutaneously. Do you know how long approximately that will persist? So how long are you expecting that implant to be in a patient? Will it require removal?
Yeah. Hi. So currently, because we are our device based on alginate, we have alginate coating for immunoprotection. So until now, we know that alginate could persist up to 9.5 years in the patient. So of course, our goal is to have the device persist as long as possible so that it could correct the blood glucose for the patient. But we have conducted quite a few interviews with patient and clinician. So we found that at least the feasible goal for this treatment to be replaceable for eyelid transplantation, for example, the device needs to be persist at least 3 to 5 years. So if the device is successful, then we expect that it could provide the glucose correction in that time span. Thank you.
I guess just a follow-up to that, are you anticipating any issues with bioencapsulation or biofilm formation that would affect the diffusion of components across that would affect the effectiveness of your treatment?
Yes. So of course we expect that there could be a problem with foreign body reaction, especially fibrosis. So our material is on alginate base. So we know that there are companies developing alginate derivative that is anti-fibrosis, and so it could be an option for us. And so I think that our concept is quite flexible with different type material. So we hope that in that case that we observe fibrosis and further foreign body reaction, then we could opt for more modified material. Thank you for your question.
Thank you. Can we move on to the next one? Kelsey and metastatic precision. Any questions from the judges?
I've got a couple of questions. One is the technology. How has it been validated that you're pretty confident that you're getting results that mimic the current diagnostic? And then it's not clear. It looks like you kind of have an arbitrary price. And my question is, what's the current diagnostic cost? And if you're saving substantial amount of time in the diagnosis, it would seem to me that in a private pay situation, which most vet care is, that the consumer would be more than happy to pay a substantial premium to get that diagnostic result much sooner than what you've described is currently happening.
Kelsey, you're on mute, I think. Try again.
Is that better? Yeah.
Yes, we can hear you.
Okay, for some reason the headphones aren't working. Yeah, so for the first question, results, yeah, so we published an article back in 2017 that confirmed that it was successful in separating the malignant cells from the cell sample and isolating them to be analyzed. I don't know if that's answering your question, but as far as comparing that to current methods, it doesn't. It will basically give you a confirmed diagnosis that yes, there is cancer, but then from there you may or may not need to do an additional screening to know where the cancer is, but for right now we're focusing on lymphoma, so we know that the cancer is coming from the lymph cells specifically, so you would know that it's lymphoma. But as far as like all cancers, we're just not there yet in the technology. Right now we're focused on lymphoma. With the price, are you talking about the price of the test to the pet owner?
Yeah, I'm talking about the price. I mean, you made some mention that you package in a box of 10 for $250.
Yeah. So,
you know, if your value proposition is as you claim it might be, it seems like you've underpriced this substantially.
Yeah, so our main goal is to keep this as affordable as possible, and making sure that the production costs are affordable so that we're profiting from it. So I think we estimated the production cost to be about 20% of what we would sell it for. So the price to me is okay. I think, and normally, so our target customer is the veterinarian. So they would be purchasing the product from us, but their customer is the pet owner. So normally when they purchase something from a manufacturer, they turn around and multiply that by 2.5. So that would cost to the owner, I mean, anywhere from $60 to $100. That's kind of to the discretion of the veterinarian. But for our cost to the veterinarian, we would package it in a box of 10 and sell it for $250. And that's just an estimate. I mean, that may change depending on when we get closer to manufacturing and we actually get some set numbers on how much these materials will cost. That could go up, but right now it's just a best estimate. And you had a couple questions. Did I get to them?
Yep, you got both of them.
Okay.
Thanks.
I'm curious as far as differentiation and other cell types that you may have looked at, for instance, mast cells in lipomas and your ability to differentiate those. That is certainly a high number of aspirates that are done in the clinic in a week. And then in addition to that, you mentioned that your secondary target was hemangiomas. And I look at that since certainly fine needle aspirant, I would think it would be limited to cutaneous and sub-Q lesions and not the more dominant heart or spleen. Can you comment on that?
Yeah, so mast cell tumors are definitely very common, and they're easy to aspirate because they're always right up just under the skin. So with hemangiosarcoma, that's more towards the phase 2 of our design. I talked a little bit about our phase 2 and how it would just give a positive or negative, and that's it would need a lot more development and a lot more research. But basically, we would be looking for target molecules and not cells. So the goal for that phase 2 would be to use a serum sample from a blood draw instead of the fine needle aspirate, and use the test to target certain molecules that would distinguish the cancer. So, for example, microRNAs are one, or nucleosomes are another. So, but that is to come with further research.
Have you looked as far as sponsorships to the Morris Animal Foundation or the Animal Cancer Foundation, both of which are not only concerned with animal health, but particularly also comparative medicine for humans?
No, I don't believe we've looked at sponsorships. Right now, we recently did the NSF, and we're going more for grants like the SBIR and the GRA, but I don't think we're to the point of looking for sponsors yet.
Can I ask you about the regulatory pathway? You seem to not be concerned about it. Isn't this a diagnostic test in a veterinary sense? Don't you need some kind of clearance through a federal agency in order to sell this product in interstate commerce?
No, so actually with veterinary diagnostics, they don't require an FDA approval. So with veterinary diagnostics, with a manufacturing company, so we plan to partner with a manufacturer company specifically for veterinary devices. So that responsibility is kind of up to them to make sure that it is effective, it's safe, it's accurate. So that's their responsibility. It's not regulated through the FDA or the USDA. But as far as our own regulatory pathway, that's going to be done through the soft launch. So that'll be a year, 2 year long soft launch where we will allow veterinarians in the real world to use our device at the same time as using current methods. And that way we can gather our own marketing data and determine like how accurate is this? Is it just accurate 50% of the time, or is it accurate 90% of the time? So that'll be our own research gathering. Right. Then, yeah.
Your false positives and false negatives and so forth. But
exactly.
Do you have this opinion that the veterinary diagnostics do not require FDA clearance? Do you have that from the FDA Center for Veterinary Medicine?
Yeah, I got it from online. If I'm mistaken in any way, I would definitely want to know more about that. But from multiple sources online and from a couple of industry people that I spoke with, that's what I got.
Thank you. Jim, we're gonna move on, and if we could ask a few questions of Benyak and piezo membrane. Sorry to interrupt.
Let's see. Yeah, I was interested in a couple of things that you mentioned in the manufacture, as well as your expectations of degradation. There have been a number of products that have been targeted for degradation in oceans and have found to be somewhat ineffective, and LPLA is certainly one of those, and particularly one that's been highly recrystallized, as you mentioned, that you process it at 105 and 160 degrees C for 10 hours. So those are things that concern me, that you may have unrealistic expectations. And you mentioned again that in your slide presentation that you're using an FDA approved PLA. The FDA does not approve materials. So that ultimately raises some concerns for me of what your expectations are.
Yes, so the FDA approval is for products that we're working on with PLA. So like I mentioned, we've been working on a biodegradable periodontitis membrane. So that's kind of where that comes from. And so once we were to make the mask, once we do make the mask and we want to get FDA approval for those masks to be used in the hospital, that's kind of what it's referring to.
There was Battelle launched a significant campaign for decontamination and sterilization using hydrogen peroxide of N95 masks. They experienced a tremendous market resistance for reuse of those masks. And in fact, it particularly failed because of that. And there was so much resistance, in fact, that practitioners wanted to get their own masks back, not someone else's. How do you begin to think that you'll be able to overcome that type of market resistance?
I mean, I'm not familiar with Battelle, but sure, it's going to be a very competitive market and trying to get into it. But right now, we think it'll be easier to get into the market because of the COVID pandemic. And as it fades out, if we're able to get into it and kind of capture the general public in its use of masks, because a lot of people are
a lot of the general public actually are using masks that are not even rated for filtration efficiency, such as surgical masks. So if we're able to grasp this customers before the pandemic leaves, and we can generate revenues to start producing masks, and show that there is reusability with the masks we've made so far. I think we'll definitely be able to maybe get a 5 to 10% of market penetration to start.
Are you considering the fact that the filtration efficiency of the mask is not simply the media, but also the fit of the mask?
Yes, and so we actually showed an image in our presentation of what our mask looks like right now, based on so it looks like a surgical mask, but it's able to offer better filtration because of the materials used and the way it filters. And we're working on creating better fitting masks to kind of increase that filtration.
Hey folks, this is Bob. It looks like we're running out of time. I think there could be a lot of interesting questions and discussion, but I'd really like the judges to finish their putting their scores in so that we can figure out who's taking home some money today. So please, if the judges can do that, thank you. So hold on for just a couple minutes. The numbers are changing as I talk. Please hold on. All right. I'm going to go with all the scores that are. Oh, there's still some motion. Sorry. All right. They're stable now, and the runner-up. Sorry. One of the judges is having some technical difficulty. I don't know if everyone can still hear me, but I apologize. So I'm getting some messages. So the runner-up is Formocyte Therapeutics, Chen Yang. So second place, $500. Thank you very much, Chen Yang, and folks from Formocyte Therapeutics. And so in first place, we have Kelsey Collins and Metastatic Precision. So thank you very much, Kelsey. You are the big winner today, and you get to take home $1,000. Thank you very much for all the entrants and all the presentations. Fantastic technologies. Thank you to the judges for all the questions. And I think we can sign off.
Thank you, guys.
Congratulations.
Thank you so much.
Thank you.
Thank you so much.